Hi viewers, welcome to SEKHAR'S Bio Factory for NEET Plant Kingdom with class Anthocerotopsida or Hornworts. If you like this video, subscribe my channel and click on bell icon to get notifications. Like, follow and share my videos to your students and friends to get more information in biology related to NEET and other national state level competitive exams. In this video, I am going to explain the general characters of class Anthocerotopsida or hornworts. Anthocerotopsida is a group. It is placed in between Hepaticopsida and Bryopsida. The Anthocerotopsida members are also called hornworts due to having of a horn-like structure of sporophyte. Most of the members, these are grows in moist shady places and sites of deattaches are in moist hollow among rocks. But a few species they grow on decaying wood. The members having gametophytic haploid plant body and it is thalloid and dorsiventrally differentiated. The thallus or gametophytic plant body, it is small, dark green, dorsiventrally differentiated with prostrate and branched or lobed. Generally, there is no midrib or spongy due to presence of underlying mucilaginous duct in this plant body. The rhizoids are root-like structures. These are simple and present on ventral surface, but tuberculate rhizoids and ventral scales are generally absent in its plant body. On the surface of thalloid plant body, some of the rounded bluish green thickened areas are present on ventral surface. It indicates Nostoc colonies. In internal structure, the internal structure is very simple without cellular differentiation. At the same time, air chambers and air pores are absent. 1 to 6 number of chloroplasts are present in each cell with oval or discoid with lamellar structures like higher plants. The antheridium are developed within antheridial chamber either singly or in group on the dorsal side of the thallus and it is the male reproductive structure. The archegonium are found in sunscreen condition means present inside the chamber at lower side on dorsal side of the thallus. Male and female gametes after its maturation these are fused to form a diploid zygote. This diploid zygote it becomes into a sporophytic structure called sporangium. This sporangium it is arises or developed from the dorsal side of the thallus and it is elongated. It is elongated and cylindrical in structure. This sporophyte or sporangium it consists of foot, meristematic region and capsule. It possesses intercalary meristem due to having of this intercalary meristem in sporophyte. It grows continuously throughout the growing season. The wall of the sporangium it contains chlorophyll pigments but at the central sterile portion is called columella. which is surrounded by sporogenious tissue and spores along with elaters are also present. Each haploid spore which is produced inside the sporangium from spore mother cell with the help of meiosis division, it can produces a gametophytic new plant body in these members. One of the example, that is Anthoceros. The systematic position of Anthoceros is it belongs to kingdom Plantae, subkingdom Embryophyta, division Bryophyta, class Anthocerotopsida or Hornworts, order Anthoceratalis, family Anthocerataceae, genus Anthoceros. Anthoceros members are common in various hill regions found in very shady and moist places. These are found in hollow on the moist rocks in the form of a dense patches. But some of the members they grow on decaying wood. The thallus or gametophytic plant body. It is a small, dark green in color, dorsiventrally differentiated with a prostrate branched or lobe having dichotomous branches. These are without midrib and spongy due to presence of underlying mucilaginous duct. The rhizoids which are developed from the lower surface, these rhizoids are generally unicellular in nature, only having a smooth walled, along with that unbranched scales are completely absent in its plant body. On the ventral side of the thallus, certain intercellular mucilaginous cavities are developed, and these cavities are open with the help of a small opening, the slime pores on ventral surface of the thallus. Sometimes the colonies of blue-green algae form Nostoc are found in mucilaginous cavities, and there is no symbiotic relationship in between these Nostoc colonies and thallus of Anthoceros. The Nostoc colonies, which may easily seen with the help of a lens from the underside of the thallus. In the internal structure, internal to the upper surface and lower surface having epidermis, and they are simple parenchymatous cells. The parenchymatous cells are isodiametrical and uniform in nature. In their internal structure, generally air chambers or air pores are completely absent. Each cell it consists a big chloroplast which possesses a single pyrenoid in its center. The chloroplasts are lens-shaped structures. These chloroplasts are superficial cells or longer than chloroplast of other cells. The nucleus it lies in the close vicinity of chloroplast near pyrenoids. Sometimes the chloroplast enfolds the nucleus within it. In Anthoceros members, the reproduction it takes place with the help of two methods. In this, the first method is vegetative reproduction and second method is sexual reproduction. The vegetative reproduction it is takes place by various ways of method, especially by progressive growth and death of thallus. Sometimes it is takes place with the help of tubers developed on its body surface. Along with that, rarely gemmae are produced on their thallus body, and along with that, persistent growing apex. These are the methods of asexual vegetative reproduction in Anthocerotopsida members. The sexual reproduction. Anthoceros, it may be homothallic or heterothallic in nature. In homothallic or monoecious species, both male and female reproductive organs are present on same thallus, but in heterothallic members or dioecious members, both male and female reproductive organs are separately present on two different thalli. The antheridium, it is either singly or in group. These antheridiums are produced in a small antheridial chambers. This development, it is completely endogenous, means the development of antheridium takes place inside of the thallus in a antheridial chamber. The matured antheridium, it is a stalked and club-shaped structure. The stalk of the antheridium, it may consist a mass of cells. The antheridium, it is covered by a single layered jacket, and inside the jacket a numerous androcytes which are metamorphosis into anthrozoids. Antheridia, it absorbs water and bursts the apical end, and it gives rise to many anthrozoids or male gametes. They move to outside of the antheridial chamber. These anthrozoids are generally in spindle-like and biciliated structure. The cilia present in anthrozoids are attached to anterior end of its body. The archegonium, it is embedded inside the thallus. They do not possess any jacket sterile cells around it. A matured archegonium, it is a flask-like in shape without neck canal cells and with an egg cell in venter. It is called oospore. The venter canal cell and the neck canal cells become gelatinous and form a mucilaginous substance. Through water acts as a medium for the transfer of anthrozoids, and these anthrozoids enters into mouth of archegonium, and these anthrozoids are attracted chemotrophically. The male and female nuclei of both male and female gametes are united to other produce a diploid zygote. It is called oospore. This diploid zygote, it is the first cell for the development of sporophytic stage. After fertilization, the zygote, it secretes a cellulogenous wall around it and enlarges in size. The matured sporangium, it consists of bulbous foot and projecting cylindrical erect capsule. There is a meristematic zone above the foot. Because of this meristematic cells, it grows continuously in this growing season. The sporogenous tissue which is present in capsule, now it is differentiated into two types of cells. In this, some of the cells are called sporocyte cells, called spore mother cells, and the sterile cells called pseudoelaters. The sporophytic cells are sporocytes, spore mother cells. It undergoes reduction division to form a tetrad of haploid spores. These haploid spores, which develops in the upper part of the capsule and mature first. Each spore it consists a nucleus and a chloroplast in its structure. The sterile cells they soon divide obliquely or transversely to produce 3 to 5 celled pseudo-elaters. These pseudo-elaters it helps in the dehiscence of the sporangium. Now the spores are released to outside. At this time the pseudo-elaters behave like true elaters. The capsule arises from the thallus in the form of a small horny structure. Because of this horny appearance, these members are named as hornworts. After their liberation from the sporangium, the spores it undergoes a period of resting prior to germination. The spores, the haploid spores, they germinate to produce a gametophytic. So Anthoceros having two morphologically distinct phases, that is haplophase and diplophase, consisting in its life cycle. The life cycle of this type, which is characterized by alternation of generation and sporogenic meiosis, is known as heteromorphic and diplohaplontic life cycle. These are the general characters of class Anthocerotopsida or hornworts. Thank you for watching. If you like this video, subscribe my channel and share my videos to your students and friends.